This conference will now be recorded.
Okay. The last class we discussed about the different types of regulations. Okay. In that, okay, we discussed about the different types of regulations. And today we are going to discuss about the regulatory enzymes. In this, mainly we have to discuss about the metabolism pathways. In the metabolism, normally we have two steps, and that are the catabolism and anabolism. I think you know all this. What is anabolism, ma? Anabolism means in the metabolism mainly two pathways are present. One is catabolism and another one is the anabolism. Catabolism means, yeah, anabolism means breaking down. Is it correct? Catabolism means, catabolism means, actually the metabolism, and these are mainly involved anabolism and catabolism. Good, Vidushi? Yeah. Anabolism is the synthesis of complex molecules from simpler ones. Good, good, ma? Yeah. Catabolism is breaking down. Asha, check it once. Okay, here catabolism is, that is the degradation of the molecular complex compounds into simpler ones, and that is called as the catabolism, whereas anabolism involves in the synthesis of different molecular compounds, and these two are involved in the metabolism pathways. And in this, in metabolism means here we have to make out the synthesis of different compounds, and also we have to degrade the compounds, means some of the enzymes are required. Mainly enzymes are required for the metabolic pathways. And here, how these are controlled, that means enzymes are mainly responsible for the control of metabolic pathways in catabolism and also in anabolism, and that is called as the regulatory enzyme. That means here enzymes are regulated in some manner, and what are the manners that are involved in the regulation, and how these are regulated in metabolic pathways, and that are called as the regulatory enzymes. Understood, ma? In the last class, what we discussed? In the last class, just we discussed about the types of inhibition processes, and that are the reversible, irreversible, in that competitive, non-competitive, uncompetitive, and that are the different types of inhibition process, how these are controlling in this. And in metabolism pathways, how these are controlled by using different enzymes, how to which extent these enzymes are regulated, how these are regulated in the metabolic pathways, and that is called as the regulatory enzymes. Understood? And here these regulatory enzymes, these are normally regulated. I think you know about this regulation process in the metabolisms, and what happens, some of the changes that occur in the enzymes, and that causes the enzyme, that means the modification occurs in the enzymes, and that causes the changes in the metabolic reaction. That means we have to stop the metabolic pathway at certain extent. That means here the enzyme has to be modified, and that is the one of the reaction. Like that, different types of processes are involved in the metabolic pathways for the control of enzyme activity, and that are called as the regulatory enzymes. And these regulatory enzymes, and these show, that means here these are showing the catalytic activity, that means increased catalytic activity and decreased catalytic activity in response to specific signals. That means here some signals are created, and that signals are attracted by these enzymes, and that causes the increase or decrease in catalytic activity, and that is called as the regulatory enzymes. Okay, and here this is the in these mainly allosteric regulation of enzyme that are mainly involved. And what are allosteric sites? What are allosteric sites? Allosteric sites means, yeah, binding site other than active site of enzyme. Good. And that means here allosteric enzymes means here these are the class of regulatory enzyme, and that are increasing or decreasing the catalytic activity in response to certain signals, and that are called as the allosteric enzymes. Here normally enzyme contains active site for the binding of the substrate other than active site. That site is called as the allosteric site. Here these enzymes are binding at that site, and that causing the change in the enzyme, and that means here these are decreasing or increasing the catalytic site by binding to a site other than the active site, and that enzymes are called as the allosteric enzymes. Understood, ma? And here these metabolic pathways, these are mainly participating in metabolic pathways. Here observe this one. Here these metabolic pathways, here these are not running in a continuous basis. These are not running in a continuous basis, and these are regulating according to the need. That means when these are needed, when the particular product and that substrate that has to be modified and that is producing the product, means that enzymes are needed, and then these pathways are running, and here two pathways are present, and here mainly catabolic pathway is there, and catabolic pathways are run if there is demand for ATP. That means here if energy is needed, that means here catabolic pathways are needed. And for example, glycolysis may be slowed down, but beta oxidation is meeting current energy demands. And you can understand this after the metabolic pathways, ma. What is I think you know glycolysis process, but beta oxidation some extent you don't know. And here glycolysis and beta oxidation, gluconeogenesis, glycogenesis, and different pathways are present, and that are involving degradation and synthesis. And here and different types of reactions, and that are present, and that is causing the synthesis of ATP and also causing the degradation of ATP. And if we have to maintain the energy, that means if we want some energy means, and some catabolic pathways run, and that causing the production of ATP, and that means here the glycolysis supports the need for rapid delivery of energy. Okay, and also different pathways that have to be controlled by using different enzyme. That means here parallelly catabolic and synthetic pathways, that means anabolic pathways, catabolic pathways and anabolic pathways, these regulations are important because we have to conserve the energy, and that means here the energy production, energy degradation, and that all are depending upon these pathways. Okay, and these pathways, and why these pathways, what is the relation between enzymes and these pathways means, here these pathways are to be controlled, means enzymes are required for the processing of these pathways. Understood, ma? Just that only I mentioned these pathways, ma. Here catabolic, anabolic, and that are the types of metabolic pathways. And here in the catabolism, we have to need, that means the ATP energy is needed means catabolic pathways or occur. Whereas ATP energy, that means here the degradation of ATP occurs in anabolism pathways, and these have to be required for the enzyme activity. And enzymes are required for the metabolic reaction. That means there is a correlation between all these types of processes. Okay, and mainly these are focusing on enzymes, and that are called as the regulation of enzymes. Mainly depends on the allosteric regulation of the enzyme. Allosteric regulation of the enzymes. Here also we have had, here glycolysis occurs when ATP, that means here it has the releasing of 2 ATP and 2 NADH molecules, and this, that means gluconeogenesis, that has cost of 6 ATP plus 2 NADH. Okay, we will discuss all these pathways in the next other classes, ma. And here that means for the requirement of energy or for the degradation, and that means here these enzymes are required, and these ought to be taking place in the metabolic pathways. Okay, and next here, and enzymes are mainly regulated in several ways. And what are the ways that enzymes are regulated? Mainly it is depending upon the allosteric regulation. In that allosteric regulation, that shows mainly of substrate affinity. Substrate affinity, that is the allosteric regulation of substrate affinity. And also enzymes are regulated by covalent modification of the enzymes. Covalent modification of the enzymes, and also by regulated expression of gene or synthesis of gene. That means here the genes means here mainly proteins are involved. For the synthesis of proteins means here what are the processes that are required? Transcription, translation, and these are the processes required for the synthesis of protein. And that means here enzymes are mainly participating in these reactions. And that means here the regulated expression of gene or of synthesis of enzyme, and these are to be regulated. And also by regulated destruction of the enzyme, and that is the main one. And also here these are the processes that are mainly required for the regulation of different metabolic pathways, in that mainly participating enzymes. Okay, and here enzymes are regulated in several ways. In that mainly 4 ways here we are mentioning: allosteric regulation and covalent modification and regulated expression of gene at the gene level or at the synthesis of protein level. Okay, and next regulated destruction of the enzyme. Some enzymes are responsible for the destruction, and that we will discuss. Okay, and here in these the first 2 effects, and these are giving immediate responses to the signals. The first 2, that means allosteric regulation and covalent modification of the enzyme, and these are giving immediate response to the change the condition. Whereas the last 2 methods, and that showing the longer response to the signals. Okay, and these are the several ways for the regulation of enzymes. And for this here in the first one, that is the allosteric regulation of substrate affinity. In that it is the main example for that is aspartate transcarbamylase, and this is a type of enzyme, and that shows the activity of allosteric regulation of affinity. And here this enzyme mainly participating in the synthesis of pyrimidines. In the synthesis of pyrimidines. And what are pyrimidines? What are pyrimidines, ma? Anyone? The nucleic acids, nitrogenous bases are present. In the nitrogenous bases mainly purines and pyrimidines are present. And what are purines and what are pyrimidines? Purines. What are purines and what are pyrimidines? Yeah, Dipshika. Okay. Nitrogenous bases. Yeah. Pyrimidines means 89 and guanine purine. Good, Vidushi. Pyrimidines. Yeah, good. Cytosine and thymine. Yeah, cytosine, thymine, and uracil. Good, Deepshika. Okay, and these are the pyrimidines. Here, in the synthesis of pyrimidines, and this aspartate transcarbamylase enzyme mainly participating. And what it requires, that means here, this enzyme participating in the reaction means it has to convert the substrate to product. And what are the products here? Pyrimidines. And what are the substrate here? Carbamyl phosphate. By using carbamyl phosphate and aspartate, here it is to be converted into the pyrimidines, and that is the here the main enzyme that is responsible here is the aspartate transcarbamylase. Okay, here this enzyme has to be regulated at some level. And how these are regulating, how these are activated means some of the steps are required for the activation or for the inhibition of the enzyme. And what are the steps? Here, ATCase, that is the aspartate transcarbamylase, that is negatively regulated by CTP. What is meant by here negatively regulated by CTP? And that means here, if the sufficient amount of CTP, that is cytidine triphosphate, and that is present, and that means if CTP is present, means there is no need to make more, and that means that enzyme is inhibited at that level, and that is called as a negative feedback control. That is called as a negative feedback control. Understood ma? Here what is happening here, it is mainly required for the synthesis of pyrimidines. That means here in the synthesis of pyrimidines, if CTP is present, and that means there is no need to make more, and that means if the CTP is present at highest level means, and that causing the inhibition of that particular enzyme, and that process is called as the negative feedback control. Negative feedback control. And next, another one is there. What is the regulation that is called as a positive regulation? And what is positive regulation here? ATCase is positively regulated by ATP. And that means ATP means here that is a purine. If ATP is abundant means here we have to synthesize the pyrimidines, and that regulation is called as a positive regulation. And that means if ATP is abundant means, and the cells has to be grown, and DNA RNA synthesis should be active. And that means here CTP levels are less whereas ATP levels are higher, and that causes the positive regulation by ATP. That means here we have to synthesize the pyrimidines, and that process is called as a positive regulation by ATP, whereas the negative regulation by CTP. Understood here? Positive regulation means here we have to synthesize the pyrimidines, whereas only purines are present in highest level here. Whereas in negative means, in negative what is happening? Pyrimidines are present in higher amount. That means here we have to, there is no need to make the pyrimidines, and that is called as the negative regulation and positive regulation. And that are the two types of regulations, and that are occurring in this process. And here this is called as a allosteric regulation of affinity. That means here this is depending upon the concentration of purines and pyrimidines, and here, and that is showing the mainly substrate affinity. That is showing mainly substrate affinity, and that is the type of inhibition called as the allosteric regulation of substrate affinity. Allosteric regulation of substrate affinity. Okay, and here, and what are the kinetics? And that are exhibited by this enzyme. Exhibited by this enzyme. Here, what is the substrate here? The main substrate here used is the aspartate. Aspartate is mainly used for this controlling of ATCase enzyme. ATCase enzyme. And here, these are represented, that means enzymes, the activity of enzymes is mainly represented in the form of kinetics. Kinetics means, and that reactions are showing in the manner of kinetic. Kinetics means here catalytic activity. Kinetics means enzyme activity in the form of catalytic activity, and that is called as the allosteric kinetics. Allosteric kinetics. And here, and this is described in terms of a low affinity T state and high affinity R state. And what are these states? Here, these are called as a tensed state and relaxed state. Tensed state and relaxed state. Here it is shown in terms of low affinity T state and high affinity R state. And what are these states? How these are occurring means here it is depending upon the substrate concentration, that means substrate affinity. That means here what is the substrate here? Aspartate. That means when aspartate concentration is low, that means the enzyme is in tensed state. Tensed state. That means it's showing the less activity of the enzyme. Tense. Tense means what happens? We are in tensed state. In tensed state means we don't that means the thinking ability of person that is decreased. Here also the enzyme catalytic activity is also decreasing in tensed state, and that is when aspartate is low means, and that causes the enzyme in tensed state. Whereas when aspartate increases, that means the concentration of aspartate and that is increased, and that means the enzyme switches to a high affinity relaxed state. Relaxing means here the catalytic reaction occurs in a manner, and that is called as a relaxed state. And here that means here depending upon the substrate concentration, the enzyme states are changing. And that means here the enzyme is normally monitored in terms of kinetics, and that are represented in the form of velocity of the reaction. And what is the velocity here? That is the maximum rate of catalysis, and that is shown in terms of V naught by V max, and which is essentially the measure of the reaction of that catalytic site. Of that catalytic state. Which substrate only? Normally the reaction rate, and that is following here sigmoidal curve. This is the sigmoidal curve. Observe this one mark, curve C, and curve C, what is happening? This is called as the sigmoidal curve. When CTP binds, CTP means here cytidine, that is present in highest level, and that means here there is no reaction occurring. That means here, and that should be in the form of tensed state. That should be in the form of tensed state. So the curve is shifted to the right. The curve is shifted to the right. That is the D1. Here this is the normal curve mark. That is the C1. C1 is the normal curve. And here when CTP is highest means, and the curve shown in the form of D1. This is the one. This is the one. Okay, and whereas when ATP is there, that means that is favoring the all state. that is favoring the OR state. That means the curve is shifted to the left side. That is a curve B. Curve B. Okay, understood ma? Here mainly we are showing three curves and that are of B curve, C curve, D curve. And what is happening here? Here actually the kinetics and that is showing hyperbolic curve normally. If we observe the enzymatic reactions in Michaelis-Menten and that what is happening up to some extent, the velocity increases and after that the velocity keeps constant. That is a normal reaction. But here in the allosteric regulation of enzyme and the graph is in the form of sigmoidal curve, that is in the form of S-shaped curve. And here what is the normal curve here? C1 is a normal curve. C is the normal curve. Whereas when CTP is more, that means it favoring the formation of tensed state. And that means the curve is shifting to the right side. That is called as a D curve. D curve. Whereas if the ATP binds means that is favoring the OR state. That means the curve is shifting to the left side of the C curve. And that is called as the B curve. B curve. Understood now? Whereas if ATP is high enough, and that means here high amounts of ATP, that means high amounts of concentrations of ATP are present, and that is not disturbing the normal reaction, and that means that is showing the hyperbolic curve. Hyperbolic curve that is indicated by A. That is indicated by A. And here for any ones here normally for enzyme the substrate concentration here for allosteric enzymes mainly the substrate concentration giving V naught by V max and that is indicated by 0.5 and that is indicated by 0.5 or K 0.5 and that means that is showing the substrate affinity. That is showing the substrate affinity. Actually, this sigmoidal curve is observed in another type of reaction that is called as the hemoglobin. Hemoglobin normally saturating with the oxygen and that's showing the reaction called as the hemoglobin reacts with the oxygen that is giving the oxyhemoglobin and if the concentration of oxygen and carbon monoxide or carbon dioxide are changing and the saturation curves are disturbing and that's showing the different reactions. Okay, in the same way here also the allosteric enzymes and these are depending upon the substrate and depending upon the substrate levels here these are changing the direction of the curve that are showing the direction of the curve and here and the substrate concentration that is giving V naught by V max that is designated as the K 0.5 here and that means here this is the modified constant of the Michaelis-Menten. This is a modified constant of Michaelis-Menten constant. Hence, it is indicated as the K prime and here K 0.5. Okay, and here also, and that means it is similar to like that of hemoglobin saturation curve, and that is showing the saturation percentage, and that is representing the oxygen saturation. Here, this is representing the oxygen saturation that is indicated by P50. Okay, and here and this is not following the Michaelis-Menten equation. That means here it is not depending upon the Km values or V naught. That means here in the reversible inhibition, irreversible inhibition, competitive inhibition, what is happening? The Km values or V max values, these are changing here. And this one here, the allosteric enzyme, these are not following the Michaelis-Menten formulas and Michaelis-Menten equation. And here the Km that is not used for the allosteric enzymes. Km is not allowed for the allosteric enzymes. Deepshika, here the substrate is the aspartate. Here, see this one here, what is happening here? This is the reaction, carbamyl phosphate and aspartate. These both are reacting with each other. Okay. And here this aspartate is the main substrate here. Aspartate is the main substrate here, and that is utilizing carbamyl phosphate, and first it is converting into the carbamyl aspartate, and later on, and that means here some of the steps are going here, and that is giving the production of pyrimidines. Okay, and that is the synthesis of pyrimidines, and mainly here it is using as a substrate aspartate. But here what are CTP and what are ATP means here, these are depending upon the concentration of CTP and ATP, and also aspartate concentration, and also aspartate concentration. Understood, Dipshika? Dipshika? Yeah. Okay. Okay, and here that means here the tends to relaxed switch, and that is mainly induced by the substrate concentration, and that is called as the positive homotrophic effect. And that means here homotrophic, what is meant by homotrophic? Heterotrophic means here homotrophic means here the T2R switch, that means here tends to relaxed switch, and that is mainly induced by the substrate occupancy. That means here only the substrate itself is involving in the reaction control or in the enzyme control, and that is called as the homotropic effect. Homotropic effect. Whereas the other than the substrate molecule is involving in the reaction means, and that is called as the heterotropic effect. Heterotropic effect. And here that means here these are mainly depending upon the substances called as the substrate. Here the substrates are involving in the process, and that causing the inhibition of the catalytic activity or increasing the catalytic activity. Here the substrates are only involving in these reactions, and that means that is following the T2R switch. T2R switch, and that is called as the homotropic process. Whereas if the other than substrate is involving in the regulation of the catalytic activity, and that is called as the heterotropic effect. Heterotropic effect. And that means here homotropic means here the change is induced by the same substrate, that is positive, because affinity is increasing with increasing the substrate concentration, with increasing the substrate concentration, and that is called as the homotropic effect and heterotropic effect. Okay, and here what are heterotropic effect? And here mainly ATP and CTP or the allosteric effectors for aspartate transcarbamylase enzyme. And what happens when ATP is present? ATP is present means in highest amounts means the reaction has to occur. The reaction has to occur, and that means here it has to be maintained in relaxed state. It has to be maintained in relaxed state, and that is called as a T2R switch, and that is induced by binding with the ATP. By binding ATP, and that is called as a positive heterotrophic effect, and that is shifting the curve left. Understood ma? Here what is happening here? The last one I said what about here? It is mainly depending upon the substrate concentration. Substrate means here only substrate, that means aspartate, depending upon the aspartate concentration here these are changing, and that is called as the homotropic effect. That means only substrate is involving in the regulation of catalytic activity, and that is called as the homotrophic effect. Whereas in heterotrophic effect, what is happening here? Mainly ATP and CTP are involved. Here, depending upon that, here that heterotrophic effect, and that is shown in the form of positive heterotrophic effect and negative heterotrophic effect. And here ATP and CTP are the allosteric effects. Okay, effectors. Here what are these? These are the effectors. And for the increasing or inhibiting the catalytic activity of ATCase enzyme. And here this T2R switch, and that is induced by binding ATP. ATP means here this is not the substrate one. Here this is not the substrate one, and that means here different substance that is involving in the catalytic activity, and that is called as the heterotrophic effect. And here what is the heterotrophic effect? Here it is observed. Here this is observed in the form of positive manner, and that is called as the positive heterotrophic effect, and that is shifting the curve left. That is shifting the curve left. Understood ma here? And this is called as the positive heterotrophic effect. Whereas here heterotrophic means the other substance that means that is involving in the catalytic activity. And whereas here tensed switch, when this tensile switch occurs, that means the increased concentration of CTP, that means cytidine triphosphate, is more in the reaction, means there is no need for the reaction occur, and that means here that the enzyme is to be in the form of tensed state, and that type of effect that is called as a negative heterotrophic effect. That type of reaction is called as a negative heterotrophic effect and shifts the curve right side, and that is shifting the curve right side. And that means here negative means here increase in concentration of CTP causing the decrease in enzyme activity, and that is called as a negative heterotrophic effect. And here these are not directly participating in the reaction, but these are participating in the reaction by the allosteric behavior of the enzyme. By the allosteric behavior of the enzyme that causing the regulation of the reaction. Understood ma? Here we are discussing about the allosteric enzymes. Here in allosteric enzymes, okay, in allosteric enzymes, mainly here these are binding at a site other than the active site. Okay, and here that enzymes are called as the allosteric enzyme, and that are showing the homotrophic effect and heterotrophic effect, and that are depending upon the substrate affinity, and also depending upon the other molecules. What are the substrate here? The main substrate here used as the aspartate, whereas the other ones here we are using are CTP and ATP. And CTP and ATP, how these are regulating in the reaction means by showing the effects called as the positive heterotrophic effect and negative heterotrophic effect. Understood ma up to this? All of you? Man, CTP is a substrate. Okay. Is it clear for all of you? Movinya, Vidushi, Dipshika, Asha, Anaga. Is it clear, ma? Yeah. Okay. And here, that means here, when both are present, what is happening to the reaction? When both, that means when ATP and CTP both are present, and that means these are competing for the same binding site. These are competing for the same binding site. And that means here the relative concentration of the two nucleotides, and that will determine the reaction that has to be happening. And that means here, depending upon the concentration of ATP and CTP, here these are giving the reaction. And that means if the CTP is, that means here cytidine has more affinity means here, and that means here, so ATP has to be significantly higher than CTP for a positive effect. That means if the CTP's concentration is more means it has to maintain the highest concentration of ATP for the reaction to occur. And that means here these are competing for the reactions to occur in. These are competing for the reaction. And here, and these are much more versatile than the simple competitive and non-competitive inhibitor, because competitive and non-competitive inhibitors, these are depending upon the rate of the reaction and also depending upon the Michaelis-Menten equation, and that is normally we are showing that ones. Here these are not following that type of reaction, these are showing different types of the reactions. These are showing different types of the reaction. And here, when substrate concentration is close to the enzyme concentration means here the positive effector and that shows the full activity, whereas the negative effector and that turning the enzyme off. And that means here these are required for the metabolic reactions to be happening. Understood ma here? If the reaction has to, that means if we want the pyrimidines means, and that means the enzyme should be active, and if the pyrimidines are highest amounts means the enzyme activity should be low, and these types of the reactions are happening in the control of metabolic pathways, in the control of metabolic pathways. And here mainly it consists of two kinds of subunits. What are the subunits here? And that are called as the catalytic subunits, and that contains the substrate binding and catalytic site of the enzyme. And that may be in tensile state or in relaxed state. Whereas in regulatory sites, these subunits are also present in aspartate transcarbamylase enzyme, and that containing the ATP-CTP binding site and also controlling the activity of these catalytic subunits by determining the tendency of T or O state. That means it has to be maintained tensive state or relaxed state, and these states and that showing in regulation. Hence, that are called as the regulatory subunits. Understood here? The enzyme mainly contains two subunits that are called as the catalytic subunit and also regulatory subunit. That catalytic subunit is the normal site, that is active site that is responsible for the binding of the substrate. Whereas the regulatory sites are the other sites than the active site that are called as the allosteric site, and that are mainly participating in the regulation process, and that are called as the allosteric sites. Okay, and that are the these are the main form of the ATCase enzyme. Understood up to this? And this is called as the allosteric regulation by substrate affinity. Allosteric regulation of substrate affinity. And next, another process that is involving in regulation, and that is called as the covalent modification. What is meant by covalent modification? Anyone? Covalent modification means? Covalent modification. Any example for this? Here enzymes. Enzymes means here these are normally in which nature these are present? Yeah, good. Phosphorylation, methylation, and that are called as the covalent modification. Okay, here what is happening here? Normally the covalent attachment of molecule, that means any molecule and that is causing the modification of activity of enzymes and many other proteins, and that are called as the covalent modification. Here normally proteins are acting as the enzymes. Okay, and here when these are modified means that the enzyme activity and that may be increased or decreased, that is called as the covalent modification here. And how this covalent modification occurs means some molecules are involving in the modification, and what are that molecules called as the donor molecules, and these donor molecules, these are providing a functional moiety and that causing the modification of the properties of the enzyme. Okay, here donor molecules are mainly involved in the modification of the enzyme by providing some functional groups, and that are called as the covalent modification. And these most modifications are here reversible modifications. Okay, and here what are the covalent modifications and what are the examples means here the modification that is occurring is here mainly phosphorylation, and here mainly ATP is providing the and phosphorylating functional groups, and for this main example is the glycogen phosphorylase, and that is mainly involving the function of glucose homeostasis and energy transduction. Okay, and next acetylation. In acetylation main molecule that is involved is the acetyl CoA, and here the modified proteins are the histone proteins, and that are involving in the function of DNA packing and transcription process. And next myristylation, and that mainly involves the myristoyl CoA, and here mainly the modified protein is the SRC, and that is mainly participating in the pathways of signal transduction. Okay, and also ADP ribosylation, and here the donor molecule that is involved is the nicotinamide adenine dinucleotide, and that is the main example for this modification is the RNA polymerase enzyme, and that mainly participating in transcription process. And also farnesylation, and here farnesyl pyrophosphate, and that is mainly the donor molecule, and that is mainly involving in the signal transduction process, and here the modified protein here is the ras protein. And next gamma carboxylation here, and the donor molecules here involved are the bicarbonate sorry bicarbonate molecules here HCO3 minus, and here the main example for this modified protein is the thrombin, and that is mainly participating in the protein function of blood clotting. Okay, and next in the same way sulfatation, ubiquitination. Okay, and these are the processes and that are involving in the modification of the enzyme. Okay, and here mainly ubiquitination, that means that is a degradation process. Here proteolytic degradation, ubiquitination. After the translation process, some of the proteins they undergo modification by using the ubiquitination process, and that is called as the protein degradation or the proteolysis process or proteasome degradation complex that is involving in this process. And that means here by different molecules, by using the different molecules here enzymes are modified, and that are mainly responsible in different types of reactions. Okay, and here in this mainly phosphorylation is mainly involved in different types of metabolic reactions. Main example glycogen phosphorylase, glycogen synthesis process and glycogen degradation process, also this phosphorylation is involved. And here in this phosphorylation and dephosphorylation, and these both are involved in the modification of the enzymes, and that is called as the covalent modification. And here this covalent modification is not readily reversible always. Here sometimes it may be reversible and sometimes it is not reversible, but some extent it is reversible, but here that means it is not easily possible. Okay, here how this means here the some proteins such as RAS or SRC, and these are localized to some phase of the plasma membrane, cytoplasmic phase of the plasma membrane, by the irreversible attachment of a lipid group. I think you know the structure of plasma membrane and like a fluid mosaic model that is mainly explaining the concept of plasma membrane. Okay, here mainly these proteins are localized to the cytoplasmic phase of the plasma membrane by the irreversible attachment of the lipid group. In this process, the covalent modification, and that is not readily reversible. This process mainly it is not reversible. And here the attachment of the ubiquitin, and that means here mainly the protein degradation process, that is the signal that is received causes the protein degradation process, and that causes the covalent modification. And also sometimes cyclins, what are cyclins, anyone? Cyclins. Cyclins. Do you know about cyclins, ma? Anyone? Cyclins are proteins, okay. And what are the function of cyclins? Yeah, proteins. Functions of cyclins. Yeah, cell cycle. These are mainly participating in cell cycle. That means here in cell cycle different phases are there, and that are the prophase, metaphase, anaphase, telophase, and also the interface is present, G1 phase, S phase, G2 phase, and these are the different phases that are occurring in cell cycle. And here the cell cycle normally occurs and further increase in cell growth, and that means here that means after the process it has to be regulated. And how these are regulated, that means the cell cycle is regulated by some of the proteins that are called as the cyclins. And here these cyclins, and these must be Okay, these must be ubiquitinated and destroyed before a cell can enter another phases. That means if the cell and that is present in S phase, or it is present in G1 phase, or it is present in G2 phase, that means the cell process is to be continued, means it has to be maintained in a regulated manner, and that is maintained by using some regulatory proteins that are called as the cyclins, and these must be ubiquitinated and destroyed before a cell can enter another process. Okay, and that means here the proceeding of the cell cycle requires the cyclins, and that is called as the covalent modification. Okay, and in this main example is the phosphorylation. In the phosphorylation mainly transfer of functional group of phosphate group from a donor to the acceptor amino acid of protein, that causing the modification of the enzyme. Here mainly the functional group is involved is here phosphate group, and that is involved in the modification of the enzyme. And how these are modified? By phosphorylation and by dephosphorylation. And what are the enzymes that are responsible for the phosphorylation? Means kinases. And what are the dephosphorylating enzymes here? Phosphatases. Understood ma? Here phosphorylation enzymes are kinases, whereas dephosphorylation enzymes are phosphatases. And phosphorylation occurs mainly by the enzymes called as the kinases. Phosphatases causing the removing of phosphate group through the hydrolysis of the side chain phosphodiester bond. Understood ma? And here, and this one is the here what is happening, observe this one ma. Here what is happening, here phosphoryl group is transferring, phosphoryl group is transferring. Here, this is the normal enzyme. This is the normal enzyme. And here mainly the functional group, that is a phosphate group, and that is acquired by means of ATP. That means here ATP is acting as a donor of phosphate group. And here it is donating the phosphate group to the enzyme. And that means here the enzyme is modified by acquiring the phosphate group. Which enzymes are responsible here? Kinases are mainly responsible. And what is the cofactor here involved? And that is the Ng+2. Understood ma? Here ATP. Okay, here ATP, and that is acting as the transfer of phosphate. That means here it is acting as a donor molecule. And next here phosphatases. What is the function of phosphatases here? These are removing the phosphate groups, and that means here the reversible process that is occurring in this type of reactions. Okay, and that is called as the phosphorylation. And the main enzyme, and that is mainly responsible for this, is glycogen phosphorylase. And glycogen phosphorylase, the enzyme that causing the releasing of glucose units from glycogen. And glycogen phosphorylase is the enzyme that is mainly responsible in the degradation of glycogen to glucose unit. That means it is involving in the glycogenolysis process. Understood ma? Here it is mainly involving in the degradation of glycogen to glucose units. Okay, and that is mainly regulated by both phosphorylation and dephosphorylation ma. Here it is mainly regulated by both phosphorylation and dephosphorylation. And in glycogen phosphorylase, two enzymes are present. 1 minute. Here muscle 1 is present, and here in this muscle glycogen phosphorylase, it is a dimer of two identical subunits. Okay, and here this is the glycogen phosphorylase enzyme, and that contains the active sites and allosteric effectors. That means it is mainly participating in the allosteric regulation, but is here, and that is mainly by covalent modification. Covalent modification is mainly involved here by acquiring the phosphate group. And here observe this one ma, here phosphorylase B is the inactive one. Phosphorylase B is the inactive one. And here the enzyme that is involved in the conversion of inactive to active, and that is called as the kinase enzyme. And what is the function of kinase enzyme? It is transferring the phosphate group. That means here it is modifying the inactive phosphorylase to active phosphorylase by adding the phosphate group. And that enzymes are here called as a phosphorylase kinases. Whereas here in the inactive to that means here the phosphorylase A, and that is to be converted into phosphorylase B. That means here, and that is removing the phosphate group, and that is maintained by using the phosphatase enzyme. And here, and this is also be inactive state. This is also in inactive state. But here, after acquiring some of the components, and this is changing into the active site, and that is called as the covalent modification of the enzyme. Understood ma? Here what is happening in the covalent modification? In the covalent modification, actually here the substrate molecules, by acquiring some functional groups, is so modifying, and that means mainly enzymes are involving in the modification. And here main enzymes are involved are the kinases and phosphatases. Okay, and here, and in this main example is the glycogen phosphorylase that contains the two units, that is called as active site and allosteric site, and that causing the modification of the enzyme. Understood ma, this one? All of you understood? Glycogen phosphorylase activity. Yeah, Vishi, okay. Manasa, Anaga. Yeah, okay, ma. And next, okay, and here this covalent modification that is occurring, but at which site it is occurring? It is occurring through the phosphorylation of serine 14 residue. At which site it is occurring, the covalent modification? At serine 14 residue. Serine 14 residue. And that converts the enzyme from a less active allosteric regulated form to a more active allosterically unresponsive form. That means here, and that is converting into an active form. Allosteric regulated form is converted into a allosterically active form. That is unresponsive form is here. That is called as active form. That is indicated by A. Understood ma here? And this is the one, and that is mainly participating in the reaction. Okay. Here phosphorylase B is the inactive ones, and here that is converted to the phosphorylase A by transferring the functional group from the ATP. That means here phosphate groups are added, and that causing the active form of the enzyme. Okay, and that is a covalent modification. Okay, another type of modification is there, and that is called as the ADP ribosylation. What is meant by ADP ribosylation? Here, the ribosylation means here, and that is mainly catalyzed by specific enzymes called as the ADP ribosyl transferases. And here, what are the substrate or used for this means here, and that using the NAD plus as a substrate, and that or mainly using the NAD plus as a substrate. And here mainly, and these are transferring the ribose groups. These are mainly involving in the transfer of ribose groups. Hence, this is called as ADP ribosylation process. And what are the type of enzymes here? These are called as ADP ribosol transferases. And here mainly the main enzyme that is responsible here is arginine ADP ribosyl transferase, and that causing the transfer of residues in proteins such as the ribose groups. Here, what is this? This is the target protein, that is a substrate one, and here by transferring the ADP ribose, okay, and this is converted into the target protein. Okay, what is happening? ADP ribosylation reaction is happening. And what are the enzymes that are responsible here? ADP ribosol, that is a functional group here, it is transferring. And that means the enzymes that are responsible here are the transferases. And what are the type of transferases here? ADP ribosol transferases here involved in this type of reactions. Okay, and this is mainly this type of reaction, these are playing a role in the toxicity of certain bacteria. And that means here modification occur. How why these are modifying means, and these should be maintain some specific functions. Okay, and here the specific functions means here ADP ribosylation, and that is mainly participating in the toxicity of certain bacteria. And here, what are the toxins that are responsible here means here diphtheria toxin, and that is causing the inhibition of EF2 elongation process, elongation factor by mono ADP ribosylation. That means these are inhibiting the protein synthesis process. And that means here the toxicity of the bacteria, and that is inhibited means at type of, at which levels means transcription level or at translation level, and that means the protein synthesis level should be inactivated by using the toxins. And that means here, and this diphtheria toxin that is inhibiting the elongation factors by using the process called as the ribosylation. Okay, and also caloric and pertussis toxins, these are provoking the ADP ribosylation of a G protein, and that causing the regulation of adenylate cyclase activity, and that is increasing the ratio of cyclic AMP and that causing the, and that means that showing the activity. And that means here different types of reactions are mainly responsible for this, for showing the toxic nature. That means here diphtheria toxins, pertussis toxins, cholera toxins. These are made in the form of vaccine preparation. How these are controlling the pathogenesis means here by controlling, that means by modifying the enzymes, and these are controlling that reaction and causing the that are involving in pathogenesis, that pathogenesis inhibiting process. Okay, and that is called as a ADP ribosylation process. And another one is that is called as the glycosylation. What is glycosylation means here? Protein glycosylation. That means after the formation of protein, that is called as a translation process. And after the translation process, the proteins are modified in different manners, and that mainly uses the process called as the glycosylation, and that is a one type of post translation modification. And in this glycosylation, mainly two types of glycosylations are present, that is called as N and O glycosylations. And that means here, and that are involving in the binding to different sites. Here, that means here amino sites are mainly involving, means that is called as N glycosylation. And that means here that are involved in the binding of saccharide chain to a aspargine or an a serine or a threonine respectively. And that is called as the N glycosylation. N glycosylation means that is binding to aspargine, whereas O glycosylation means that is binding to a serine and threonine sites, threonine residues. Okay, and here N-glycosylations occurs on aspargine, and that is belonging to a sequence called as the aspargine. That means here the aspargine must be the first amino acid. Okay, and that can be any amino acid that in between these amino acids. And orthoglycosylation, these occur on hydroxyl groups of serine and threone residues of side chains. Okay, and that is called as the glycosylation process. And here, what are the enzymes that are mainly involved here? Or glycosyl transferases. Glycosyl transferases, and that involving in the addition of galactose residues from the UDP galactose to N-acetyl glucosamine, and that are attaching to the proteins by different linkages. And here, and these will come in translation modification of proteins, orthoglycosylation and N-glycosylation, and these are the main processes that are involved, and that are mainly involving in the regulation of the enzyme. And what are the enzymes here mainly responsible means glycosyl transferases, and that means these are mainly involving in the regulation of different metabolic pathways, and that is called as the glycosylation process. Like that, many covalent modifications that are occurring. And here another one is clear, that is called as the adenylation. What is adenylation here means? That is binding of a adenyl group, by binding of a adenyl group to tyrosine residue of protein, and for this main example is a glutamine synthetase, and that causes the condensation of ammonia with glutamate, and that is giving the glutamate. That means here adenylation, there is a covalent modification. Here adenylated groups are adding to the enzymes, adenylating groups are adding to the enzyme. And here, and this means adenylyl transferases that are mainly involving in these reactions, and that involves the phosphodiester bond between the hydroxyl group and tyrosine group in glutamine synthetase and the phosphate group of AMP nucleotide. Okay, and this is another process that is involved in the covalent modification. And in this mainly DNA ligases, RNA ligases, these are mainly involving in the formation of phosphodigester bonds. phosphodiester bonds, and at single strand breaks with adjacent 3' hydroxyl and 5' phosphate terminate in DNA or RNA respectively. And here in this, the main process that is involving in this here is a adenylation process. Adenylation process, and that is mainly to the active site of amino groups of lysine residues. Okay, and that means here main enzymes that are catalyzing this formation of phosphodiester bonds are DNA ligases and RNA ligases. And what is the function? That means what is the significance of these covalent modification? Because the proteins has to be function means this has to be modified in a certain manner. And that means here the conversion of enzyme from one form to another, and that is converted by covalent modification. And that causes the change in activity of the enzyme, and that causing the amplification of the initial signal. Okay, and these are the reversible modification. That means these reversible modification, and that are controlling the metabolic pathways, and that means the signals are received by the metabolic signals are controlling these pathways. Okay, and that means when the stimulus is removed means, and that is converted back to the resting state. That is called as the covalent modification. That means that is involving different processes: adenylation, phosphorylation, ribosylation, and these are the different processes that are involved. Understood, ma? And we will continue this in the next class, ma. Okay, and this is the regulatory enzymes. If you have any doubts, you can comment me, ma.
Okay, thank you, ma'am. So it was really very informative. So students, if you have any query regarding today's topic, please comment. I need an answer from everyone. Please comment. Okay, no doubts. So if you understood today's topic, today's stuff, please comment yes in the comment box. Just let us know. Okay, thank you everyone. Thank you for support. Okay, now you can leave the meeting. Thank you.